You're most welcome to today's talk, and we're going to focus on the Oxford AstraZeneca vaccine and some pretty good news that's come out about it. So let's try and make sense of this. But just in case you're short of time, I'm going to give you some background information on it briefly. This is the bottom line. Now, if you give a single dose of the Oxford AstraZeneca vaccine, the efficacy between 28 days and 90 days after that single dose is 76% protection against infection. So that's pretty good. No one was hospitalised after 22 days after a single dose of the vaccine. So it seems to be protecting against severe disease, which is really good. Now, they also tested people with antigen studies to see if they were testing positive or not. And they found out that in the vaccinated group, 67% less people were testing positive. And if they're not testing positive, of course, they haven't got the virus and they can't pass it on. So from that, we can say that after a single dose, transmissibility is reduced by 67%. So the single dose of the vaccine is preventing people spreading the virus as well as protecting them and keeping them out of hospital. So pretty good news. But of course, we have to give the second booster dose eventually. And if you give that after 3 months, so the combination of giving a booster dose on day one and then 3 months later giving the second dose, the booster dose, the efficacy after the booster dose is 82.4%. 82.4%. And again, no one was hospitalised in that group after the second dose of the vaccine. So it really is quite remarkably good news. Now let's go through that now in a little more detail to work out where this has come from. Now this is the Oxford vaccine group. So that's their publication there. But the main publication is in this Lancet preprints. And this is the reference for that. The full paper is there. It's all available. So single dose administration and the influence of timing of the booster dose on immunogenicity and efficacy of the Oxford vaccine. And again, just to give you a bit of a bottom line here, really, it's not so much the dose that's critical, it's the timing between the doses that has shown to be so important in promoting the amount of immunity that the virus gives. And these longer intervals between the primary dose and the booster dose are enhancing the immunity. So simple finding, but really quite profound and really big differences between the groups, as we'll see. Now, the background, as you know, for this, this vaccine has been approved for emergency use by the Medicines and Healthcare Regulatory Authority. In the UK, we're giving 2 standard doses, not the lower dose anymore. That's finished now. 2 standard doses, 4 weeks to 12 weeks apart, but we're trying to give it towards the end of that 12 weeks, and that's where the data is showing it works well. And this is a pooled analysis of all the trials and data collected so far. So it's a bit of a review of everything that we know about this from the data so far. Exploratory analysis, immunogenicity, the degree to which it generates immunity, and efficacy for extended time intervals between the priming dose, that's the first dose, and the booster dose, that's the second dose of this vaccine. And this data is going to look at data after, well, we're going to look at a bit of both actually, but data after 1 dose is the key thing. Is it working after 1 dose? And as we've said, basically it is working remarkably well after 1 dose. Now, the method, there's a phase 3 efficacy trials in the United Kingdom and Brazil. Now, these are ongoing. So when the initial trial finished, the initial data was collected. They didn't just stop collecting data then. They went on collecting data now. So the initial data was enough to give emergency approval, but now we know that much more because more time has passed and more data has been collected, as we knew it would be. So phase 3 efficacy trials in the United Kingdom and Brazil. Phase 1-2 clinical trials in the UK and South Africa as well. So that data has been taken into account from the phase 1 and phase 2 trials. Now this data goes up to the 7th of December. Now it's taken them a while to analyse it, but this data was collected up to the 7th of September because there's always going to be this delay in analysing the data. People over the age of 18 were recruited into this trial, and they're randomised 1-to-1 into the experimental group who got the vaccine and the control group who did not get the vaccine. Blinded independent endpoint review committee analysed the data. So in other words, a committee that was blinded, in other words, they didn't know which data came from which group, reviewed the data. So this data has been thoroughly checked. There we go. Thoroughly checked by this external adjudication group as well. Now, the participants, we're dealing with fairly large-scale studies here, so it's good. 17,177 baseline seronegative participants. So they were actually tested to make sure they were negative before they went into the trial in this particular trial. UK, nearly 9,000. Brazil, 6,753. South Africa, 1,476. So a good cohort. And also this is good because we're getting people from different countries. So the result can be much more generalised. It's a fairly internationalised study. Now, infection after the first dose. This is what we need to go with. So positive infections. People diagnosed positive, 619. That's out of this original whole cohort. So they were diagnosed positive. Symptomatic infections after 14 days after the second dose, and the number there was 332. Now, initially it was 131 when the data first came out. So now they've added another 201 positive patients to this. So we can see we've got a lot more data here to be working with. This is really quite a significant update. Now, primary analysis of overall vaccine efficacy after 2 doses. Now, this is based on the original data, and it was found out after 2 doses to be 66.7% based on the original data. But of course, that was only based on 131 people that were diagnosed positive. That number has now gone up to 332. So we've got much more data to be going on here. Now, in the vaccine group, after 2 doses, there were no hospitalisations. Excellent. And there was 15 in the control group. So that was looking good after 2 doses. So we knew that people weren't getting sick after 2 doses. But of course, are people getting hospitalised after 1 dose is the big question with this UK strategy of just giving 1 dose. So hospitalisations after a single dose. Now, if it was less than 22 days, there was 2 hospitalisations in the vaccination group and 7 in the control group. So while the vaccine was working during the first 3 weeks, it was still possible to be hospitalised and have had the vaccine in the first 22 days. But after 22 days after the first dose of the vaccine, no one in the vaccination group was hospitalised. And there were 6 in the control group hospitalised. Now, of course, this is still based on a relatively small sample size. As we vaccinate millions of people, then there may be some hospitalised. But so far it's looking good with 0 people hospitalised 22 days after the first dose of the vaccine. This is pretty impressive and quite a strong vindication for the single dose policy that the UK has been taking. So looking good on hospitalisations 22 days after the first dose of the vaccine, looking very good on that. Vaccine efficacy after a single standard dose. Now, this is after giving 1 dose. From day 22 to day 90, when the second dose would usually be given, efficacy was 76%. Now, this is really quite interesting because we notice that the original efficacy was 66.7% in the first trial. So we see that the efficacy now, after 1 dose, now we have larger numbers, is actually better than the efficacy was after 2 doses. So again, remarkably good news. So from 22 days to 90 days, 76% protection. 76% efficacy against protection against infection and symptomatic infection. And as well as that, it just keeps on getting better. Protection did not wane during this initial 3-month period. Now, it did start tailing off a little bit towards the end of the 3-month period. And the paper actually says that if you didn't give a second dose after 3 months, the immunity would gradually start waning off. So this second dose is needed, but 3 months is okay. But we won't want to leave it 4 or 5 months, but up to 3 months is okay, basically is what this is saying. Antibody levels were maintained during this 90-day period. Excellent. So after the first dose, antibody levels were maintained while we're waiting for the booster dose. And minimal waning by 90 days. So it did start dropping off a little towards sort of 85, 86, 87 days. And it did drop off, the antibodies did drop off after 90 days. But up to 90 days, they were still long enough to give this high level of efficacy, this 76% efficacy. So great, that's the efficacy after a single dose. In other words, we can easily wait for the 3 months. But it actually gets slightly better than that even, because the longer gaps improve the overall efficacy. As we said, it goes up to 82% afterwards. So it actually gets better. So longer gap after 2 doses. So efficacy with a longer prime boost interval. So between the first dose and the second dose. The vaccine efficacy then was 82.4% if there was a 12-week gap, after a 12-week gap. So that is pretty good. That's after the full dose. Now, how long this level of immunity lasts for after the second dose, of course, we don't yet know because the time has not elapsed. But the data is looking good so far. So if you have a shorter dose, so basically that is the, you get 82.4% efficacy if you wait for 3 months between doses. But suppose you give the second dose less than 6 weeks. This is a second group. Then if you give the second dose less than 6 weeks, the longer term immunity that you're left with after 2 doses is only 54.9%. I mean, this is just an amazing difference. So if you give the first dose and wait for 3 months, and then you give your second dose, the immunity you end up with is 82.4% efficacy. Brilliant. But if you give the first dose and then you rush things and you give the second dose less than 6 weeks after the first dose, your efficacy goes down to 54.9%. It's a completely striking, dramatic difference. So what's emerged in the study of this Oxford vaccine is it's not so much the initial dose that was given, because you might remember initially giving a lower dose and then a higher dose seemed to have better efficacy. But when they've gone back and looked at that, what they found out that was the timing between the doses was much more significant, way more significant. And this is a huge difference. So wait 3 months, get 82.4% efficacy, rush it and give the second dose within 6 weeks, go down to 54.9% efficacy. So which efficacy would you like? 82 or 54? Well, obviously we want to wait for the longer time. An impressive difference, a striking difference. Now, this is based on who actually gets the infection. This is sort of based on the epidemiological data. But is it supported by the lab data, the immunological data? Well, observation supported by immunogenicity data. Yes, it is. Binding antibody response more than twice as high after an interval of 12 weeks. So in other words, this group had twice as many antibodies as this group. If you wait for 3 months, you end up with twice as many antibodies as if you rush it and give the second dose in less than 6 weeks. Binding antibody response more than twofold after an interval of 12 weeks or more. Impressive. Compared with the interval of 6 weeks or less. Now, this is only based on data from 18 to 55-year-olds. And this has caused a bit of confusion in France and Germany in places where they're only authorising the vaccine for use in under 60 or under 65s. Anyway, they're worried about that. But actually, we looked at the phase 2 data, which this takes into cognizance. And with the phase 2 data, the immunogenicity in the over 65s was almost as good, or pretty well the same, as in younger age groups. So I don't think they need to worry about anything, but based on this data we have here, that's only for that 18 to 55-year-old age group. But we believe with time, the efficacy in older age groups will be substantiated as well from the data that we have from the phase 2 clinical trial. So this interval between the two vaccines is just making a huge difference, as well as buying us the time, which was probably mostly the original intention because we're so desperate to get this vaccine program out. Right, interpretation of this data. What does this mean? UK single dose strategy, booster at 3 months, that's what we're doing. So direct quote from the paper, is an effective strategy for reducing disease. Reducing disease. It reduces people getting the disease, it reduces hospitalizations, and it reduces spread of the disease. So we've answered that question now. Are these vaccines helping to reduce spread? Yes, they are. And it may be optimal for rollout of a pandemic vaccine when supplies are limited or short, but it looks like it's giving us better data and better results anyway. It is the dosing interval and not the dosing level which has a great impact on the efficacy of the vaccine. Longer prime, so longer gaps between the first and the second dose, between the prime and the booster intervals, done with other vaccines such as influenza, Ebola and malaria, yielded similar results. So what we're saying is this has been observed before with vaccines being developed for influenza, Ebola and malaria, so it's not entirely surprising. These results aren't entirely surprising, and indeed they were hinted at quite clearly as early as phase 2 of this clinical trial when we looked at those some months ago now. Now, Professor Andrew Pollard, co-author, chief investigator of the Oxford vaccine trial, these new data provide an important verification of the interim data that was used by more than 25 regulatory authorities, including the Medicines Healthcare Regulatory Authority in the UK, the European Medicines Agency, to grant vaccine emergency authorisation. So this has been vindicated. This is agreed with. It also supports the policy recommendations made by the Joint Committee on Vaccination and Immunisation in the UK for a 12-week gap between the prime dose and the booster dose, between the first and the second dose interval, as they look for the optimum approach to roll out, and reassures us that people are protected from 22 days after a single dose of the vaccine. Remember that the data we have so far shows that no one was hospitalised 22 days after their first dose. Now, in all this good news, it is absolutely vital that people that have had the first dose or the second dose of the Oxford vaccine do not behave any differently until we are officially told that we can. Well, when I say we, I haven't had any doses yet, but until we're officially told that we can. So this shouldn't influence behaviour yet. This is not yet peer reviewed. It's not yet government policy, but it is remarkably encouraging, but it must not change behaviour yet. This is a preprint study, therefore must not be used to influence policy making and clinical decision making in any way, shape or form. It's not peer reviewed. I've had a good read of it and it looks pretty good to me. Not that I'm saying I'm a peer with these guys, far from it. But it's looking good so far. And finally, the vaccine, people are less contagious after a single dose. So an analysis of preliminary chain reaction swab tests in the UK. So they carried on testing people. Vaccine may substantially reduce the effect on transmission of the virus. Now, they say that because people that have been vaccinated, they had 67% of them tested negative compared to people that were in the control group. So there was a 67% reduction in positive swabs amongst those vaccinated. 67% less positive tests than you would have expected from a random sample of the population. And of course, if that 67% is testing negative on the PCR test, that means they haven't got the virus. And of course, if they haven't got the virus, they can't transmit it. So although we don't know this from full epidemiological data yet, it's looking like after a single dose, we're going to get 67% less transmission of the virus, which is a nice bonus finding. So there we go. All pretty encouraging. After a single dose, the efficacy is 76%. No one hospitalised after 28 days. And they are also 67% less infectious after a single dose. And if we wait for 3 months after the first dose before giving the second dose, instead of getting about 54% overall efficacy, that leaps up to 82.4% efficacy. Quite amazing increase in efficacy, all due to this interval. So pretty good news on the Oxford vaccine for the UK. Pretty good on the Oxford vaccine and news anyway, as this is going to be rolled out. It's being made in India, it's being made in other countries. And as I think I said quite a few months ago on this channel, actually, I expect this to be the main vaccine that the world uses to generate global herd immunity. All the other vaccines will join in, but I think maybe in 5 years' time, when we're looking back on this, it may well be that the Oxford vaccine was the one that was used most. Cheap to produce. AstraZeneca are making it at no profit whatsoever. It's being made at cost price. It really is quite incredible. It costs about $3 per dose. It can be transported in a normal cool box at normal fridge temperatures. And we know the safety profile is good as well. What's not to like? It really is looking remarkably encouraging. So be encouraged, but don't change your behaviour for now. Thanks for watching.